Ushma Neal here with the next in the Journal of Clinical Investigation series, The Conversations with Giants in Medicine. I have the distinct pleasure of speaking with Dr. Craig Thompson, President and CEO of Memorial Sloan Kettering Cancer Center. Thompson is a clinical hematologist-oncologist and immunologist who's taught us much of what we know about the fundamental ways that cells survive and replicate. Cells from immunological cells like T and B cells to stem cells and cancer cells. This interview is particularly entertaining for me since when I'm not working for the JCI, Craig is my boss. So thank you very much for joining me here today.
Well, it's great to be here, Ushma, and you're right, this is a different situation than our normal interactions at work, but it's great to be here.
Thank you. So for those who might not know you as well as I do, can you tell me a little bit about where you grew up, your parents, what you were like as a kid?
I grew up as a military brat. My dad was a career officer in the Coast Guard, and so I spent my life growing up on half the time on military bases and half the time in new cities that had Coast Guard bases every 2 to 3 years. And so it was a wonderful upbringing. Many people asked, didn't you worry about the stability? But no, it always led to new situations and new opportunities. It may be partly why I enjoy science so much. There's always a new question and something new to do. But we started out life in the Midwest, moved to Boston and the East Coast, had my first science experience at Woods Hole Biologic Institute when my dad ran the Coast Guard base in Falmouth, Massachusetts. I learned to swim at the Woods Hole Biologic Laboratories beach and had my first science lecture in first grade there, to moving to Hawaii and then many other parts of the United States over those years. And so I think became quite open to new situations and new opportunities as a result of my upbringing.
What were you like as a kid? Were you a good student? Were you mischievous?
I was not the best of student when I was in elementary school. In fact, if we hadn't moved when I was going to second grade, I probably would have been held back because I was not a very attentive young man. The good news for me is I was always pretty good at math, and so that always carried me through, but I was a horrible reader. And so virtually every time we moved, I would start at the lowest part, because back then the way they placed you in schools was by how well you read, and I was almost always doomed the first day of school. The teacher would bring you in front, give you a book, and you read out loud to the class. It was an absolute disaster. So I almost always ended the day in the back of the room in the D study group.
But at some point in time that changed, because I know I've heard you say you were editor of your high school newspaper.
So fortunately, you always have breaks in life, and when we moved in the middle of my 5th grade, up until then I had always been in the worst group for studies. The teacher read my transcript and gave me one of those classic tests that I think they still use to today, where you have a learning comprehension. You read a little story, you might answer 4 or 5 questions, you read another little story. I had never, ever, because I was such a slow reader, ever finished one of these. And the 20 minutes are usually allotted for these tests they used to do in elementary school. And the teacher brought me in. It was the middle of the year, and I was put off in an abandoned classroom, and she forgot me for an hour. And for the first time, I actually read all the stories and answered all the questions. And immediately, it was just what I said earlier, we went back into the room. I remember it vividly. She made me read to the rest of the class. I was placed immediately in the D study group and would be reading from workers that were really below grade for the rest of the time. And at the end of the day, she came up to me and asked me if I had really answered all this by myself or if somebody had helped me. And I think, unfortunately for this interview, because I've told you some of these stories before, the story as it played out was that she became convinced that I had actually answered it myself and had done better than her best student because I'd actually read it, or they had an hour to think about it. And then she started to ask me what books I was reading on my own, and I mentioned that I had just finished a book by Mark Twain called Pudd'nhead Wilson. And she immediately then started to think I was maybe confabulating a little, because she had never written a book like that. And you must mean Tom Sawyer, Huck Finn, or something like that. And I said, no, I just finished this book. I read all those. I was reading this book called Pudd'nhead Wilson, and she went to the library that night to discover that there was actually a book by Mark Twain. And after that, she put me into a reading group where I didn't have any time tests. I was allowed to read whatever I liked to read and write a book report on it, and that I could do. I might be slow, but I could actually read. And so from that, when you're not held back, you get into the better groups. And so by the time I got to junior high, I was in a situation where learning became fun, and I wasn't in the group that everybody in class made fun of. And it became interesting to participate in schoolwork. And so by high school, I was taking challenging courses and having a good time, and challenging myself to learn the things I didn't know how to do. I was terrible at public speaking. I was terrible at writing things that other people would read.
But at some point along the way, you also kindled some sort of an interest in science, or did that not happen until you got to college?
I was really fortunate in high school. We moved again when I was in high school. Just as I entered high school, we moved to outside Boston, Massachusetts. And my parents did at that point knew that education, for both of them, they were first in their generation to go to college, and so they were very appreciative of having gone to college. And so they put us in a school in Wayland, Massachusetts. It's a public school, but it had been funded by the Ford Foundation. And it was an experimental school, and we were hooked up to MIT. So the math and science classes had the first computer connections back when computer was time sharing there. And as I said, the one thing I had always been good at was math. And so I got put in because you got to have a special homeroom if you wanted to do the computer homeroom. And that got me exposed to the science nerds of the school. And it turns out I was one. And we had a great time through high school, hanging out as a group of us. Because of that situation, the science building had its own building. The teachers were dedicated to that, and we had a homeroom there so we could always come back. So it was that opportunity, and almost all of us that were in that group went into science in some way. Because we moved so much when I grew up, one of the ways you learn as a military brat to break in is that sports are the leveler. You can always join in if you know how to play the sports. So I had always gone out for sports teams and played on sports teams, and I'd played several sports in high school. And as a result, I had the opportunity to go play sports in college, and the best opportunity for me was to get a scholarship so that I could go to an Ivy League school. But the reason the Ivy League schools wanted me is because I could play sports. And so I was lucky enough to go to Dartmouth and play 2 sports there, which were the 2 sports I liked best in high school. This sounds odd, but I played soccer in the fall, and I did whitewater kayaking in the spring, which had just been made an Olympic sport, and the Olympic coach was at Dartmouth. And I had done that in high school because I had destroyed my knees as a sophomore and couldn't play baseball any longer, and so I'd taken up kayaking because my dad had grown up in northern Minnesota where that was a big sport.
So when you were in college, at some point you decided to go towards a medical degree and finished a bachelor's in medical science at Dartmouth and then transferred to Penn for medical school. So what made you decide that instead of following your father into military service as an officer, that you would go for a medical degree?
It had become clear through college that I enjoyed sciences and I was the best at performing at sciences, particularly when it got to laboratory sciences. I loved working in the chemistry lab and doing projects there. And that led me to think I should probably go on and get a doctoral degree in chemistry. That all took place when you start to think about, gee, this is going to end and I have to do something. In 1974, the National Science Foundation stopped giving out doctoral fellowships in chemistry. It was one of those times. Everybody thinks today is the only time funding for federal government withdrawn, but they decided we had enough chemists and they weren't going to fund any chemistry. So my very closest friend, who I had worked with in the laboratory, I'd worked in one of the professor's laboratories, who was a first-year graduate student, had lost his funding. I had been thinking about applying to an organic chemistry PhD program to find out I'd have to find some way to pay for it, not the way we do it now, and actually it had been done before at that time. And I was actually disconsolate about this when one of the guys on my floor in my dorm said, I'm going to a pre-med meeting tonight, you should come along. And I had never thought about going into medicine. I fainted at the sight of blood and wasn't particularly interested in medicine and biology at that point. But I went and heard this There were several people that had come from different medical schools to explain what the process of getting in, but also what you did in medical school. And I realized that was the only profession that I actually knew that engaged in science, or thought I engaged in science, because the only person I know as a scientist growing up was the base doctor. And that seemed like a perfectly good way. I didn't want to go in and follow my dad's footsteps in the military, but it seemed like a job I knew what people did, and it had a good service component, which is why my dad had stayed in the military, and particularly in the Coast Guard for the service component. And so I decided at that meeting, for no good reason, that I probably could apply to medical school and I thought I could get in. And that was incredibly naive, and I only applied to one medical school. And I fortunately got into that medical school, so.
Now, after medical school, the Navy paid your way through medical school, so how did you go about fulfilling your military obligation?
I became very entranced by intermediate metabolism in medical school and started working in a laboratory, and realized that we really didn't know very much about the science of medicine. And from that became very interested in the academic pursuit of medicine. That isn't what the Navy thinks they have a scholarship for, and quite rightly, they needed people that would actually take various service jobs to take care of the workforce that's in our military. In the case of the Navy, it's on the larger boats, the aircraft carriers, the destroyers, as well as the base hospitals, which I was familiar with. And I realized that would probably not do much to advancing an academic career. And so in the middle of medical school, I started exploring whether there are any opportunities in the Navy, and went and visited all the naval hospitals that had training programs, and ended up at the end of every visit telling them that I really thought they would be better if they let me train in academic medicine. And then I came to the Navy, where I could actually be useful in terms of what they needed in their specialty services. I somehow luckily convinced the leaders of the 4 major training programs in the Navy that that might be true, and so I was able to defer my Navy commitment and go to the Peter Brett Brigham Hospital in Boston to do my internship and residency. And at that point, it really sealed. I finally met a group of people that are at this meeting and thought exactly the way I did about the future of medicine and science, and realized this is what I really wanted to do in life, but I had a 4-year obligation to do something else. And for all the bad things we know about Harvard, Harvard has some really good things too. And so I went and explained this to the people who were running the house staff program at the Brigham, that I really didn't want to go and be an internist who worked in a base hospital, but I wanted an opportunity. Everybody back then was concerned about the Vietnam War, and there was the opportunity to go to the NIH as a researcher. That wasn't possible, but the Navy and the Army also had very good research programs at the time. And many docs, although we always know the stories about the NIH, had done equally good service jobs in the research arms of the Navy and the Army. So learning, and I was connected with several of the people that had had very good research experiences in the Navy, I realized that that would be something I really would like to do, and I thought I could excel at. And that's where being brought up in the military helps. You know that every government has a loophole, so I investigated where I could be assigned once I finished my deferment for my clinical training. And this may sound terrible to your interviewers, but what I learned is, I know one thing about the government, it is if you don't finish a program, you're considered a failure. So I worked out the opportunity to leave my residency in my senior year, apply for an exemption, because back then we were the first class that had to do a senior year to get internal medicine residency training, to apply an exemption to get internal medicine boards at some time in the future after I had more experience, and enter the military a year early. Since I hadn't finished my internal medicine training, they couldn't slot me into the plans, and the Navy has a clear path for people like that. They put them in the research division. So I was given
Ideal.
not many opportunities, but a really good one came up, which was to work at the Naval Blood Research Laboratories in Boston, Massachusetts. That's a really important place that almost no one knows about in terms of medicine. It's an important place because after World War II, military medicine advanced. We all know the stories of the vaccines that were developed around various military campaigns that the U.S. Was engaged in. But World War II was a transformative war for a horrible reason. It was the first war in the world where more people died of bleeding to death from the projectile industry, being shot, than from the gangrene they got or from the infection they caught by marching through swamps. And so for the first time, it mattered that you could resuscitate people on a battlefield with blood products. And the real problem was there weren't enough blood products whenever a battle broke out to resuscitate the number of people that were at the moment needing blood products and blood refusion. And so after World War II, the Naval Blood Research Laboratory was founded. It made sense that that would be in the Navy because the Navy was the one that had big enough ships to carry blood products around the world, so they could be the blood banks of the world. But you couldn't keep blood for longer than 35 days. You couldn't know that you were going to have a battle or there was going to be a war in 35 days. So the obvious solution when people sat in a room and thought about it is we had to figure out how to freeze blood so that blood could be resuscitated, is actually what they called it, and then given in a short period of time to somebody bleeding out, but otherwise in a freezer, hopefully for years. And so that was the mission of the Naval Blood Research Laboratory when it was founded immediately after World War II. The Korean War turned out to be the first perfect test of that. They are the ones that figured out how all the protocols everyone uses to freeze blood cells in the world, and all the cell lines that you've frozen in the world, and everything else about frozen cell products. They worked all that out over the next 7 or 8 years, and then they practiced it during the Korean War.
And what did you study while you were there?
So my job was interesting, my actual job. So I don't want to think that you just get out of a service job. My actual job in the billet that I occupied in the Navy was twofold. My job was to take care of 37 freezers, big Revco freezers that were kept at minus 140, and were storing units of blood that had been collected from the Korean War through the Vietnam War. And my job was to bring people back in every month and transfuse a certain random number of these units after we'd radiolabeled them with chromium to see if they actually functioned as red blood cells and circulated and carried oxygen. And so I carried a beeper all the time that I was at the unit, where anytime one of these freezers, and this is back when freezer technology was great, so there were many nights of running in and figuring out how to get these units into another freezer. And my second job was that I was a backup officer. All nuclear submarines require during times of war a physician, but they do not have them during times of peace patrols. So I had a billet on a nuclear submarine in Groton, Connecticut, and I would have to practice driving down there if we ever got into a war and jumping on a submarine periodically to fill out my other job. That doesn't occupy all your time, so fortunately they gave me the opportunity to hire a technician, and I could work on any medical problem I found interesting. It had to be in blood products, so I worked on why it is that we need a platelet count of 50,000 to have normal coagulation, but our normal platelet count is 350,000. And so I worked on platelet physiology, and I pretty much, with some help of some really great people who were willing to take me under the wing, learned how to do experiments, design things, and carry out experiments. So I had a great few years. I moved to the Naval Medical Research Institute in Bethesda, Maryland. I was a general medical officer at the National Naval Medical Center, which was a great job for training as a general internist and attending on the wards. At the same time, I now had my own laboratory, and I was learning how to do immunology research. In the middle of all that, the military decided that they needed to have, because at that point the Navy controlled 250 nuclear reactors. There was increasing concern in the world's community that there might be a nuclear reactor accident, and what would we do about that? And the military realized that bone marrow transplantation was now working, and that they needed somebody trained in radiation injury and bone marrow transplantation. And they offered the opportunity to start a unit at the National Naval Medical Center where they would do bone marrow transplantation on an emergency basis, and they would train a few of us. So this is what doesn't make sense. And so anyone listening to this should not necessarily think I'm very logical in how a career. I agreed to to take on another 4 years of obligation to the military in exchange for being sent to the Fred Hutchinson Cancer Center to do a fellowship in bone marrow transplantation and hematology oncology, which is what I did.
And you trained with Donald Thomas.
And I trained with Donald Thomas, which was an unbelievable experience. Don was the most consummate clinical researcher I have ever met.
Now, I've heard you talk before about, when you first started, seeing the patients just wither away before your eyes,
Right.
and the amazing transformation you were able to see through the technology of bone marrow transplantation.
Right. So I think that was an eye-opening experience. At the time I went to the Hutchinson Center in the early 1980s, we still didn't have the modern immunosuppression, so graft versus host disease for many of the patients was a horrible disease. Most patients still had their leukemia come back because we hadn't figured out all the conditioning regimens. So there were two really amazing experiences as part of that. One was actually the opportunity to see somebody who had had truly terminal illness come back from the grave, because 25% of the patients we treated truly had miraculous recoveries, went back to their normal lives. The second was the ability to deal with the most significant side effect occurred during my first year as a fellow. And I was lucky enough to be one of the fellows that treated the first 100 patients with cyclosporin A for bone marrow transplantation. Cyclosporin A had already been used for solid tumors, but Don enrolled in a protocol where we would actually use cyclosporin as the immunosuppressant. Before that, we'd use conditioning with toxic drugs like methotrexate. And it was transformative in terms of how well the patients did. And it really made bone marrow. There are many advances that have been made since then, but in my career, that was the one that transformed the disease from a purely experimental procedure to something that could be practiced everywhere.
So how do you transition from that kind of an experience that's so impactful on a clinical side to becoming an immunologist, which was sort of the first stage of your independent research?
So as I told you, I became interested from my experience at the blood research laboratories and immune cells and lymphocytes, and went and got 2 years of training in an individual named Erwin Scheer, who had a laboratory at the Naval Medical Research Institute. It was there I met Carl June, who was a chief medical resident the year I was a junior attending. And when they asked people to raise their hand to go out for training in hematology oncology, Carl and I were the two people that raised. So I went out with Carl, and we were fellows together all through the time of the Hutchinson Center, and we asked ourselves exactly that question in our second year. And both of us were interested in immunology, and both of us were interested in these patients we had just taken care of that had been dramatically advantaged by cyclosporin A. And to us, that was a great question that nobody in immunology was going to answer, which is cyclosporin had been tested in all basic science models. It turned off the proliferation of every known in vitro assay of lymphocyte function. And yet, when we were fellows on the ward and starting to treat these patients, we're going, well, that's really great. They won't have graft versus host disease. But the other nightmare of taking care of these patients in their first 100 days, and they were all required to live with us in a literally a apartment building right next to the Hutchinson Center for the next 100 days until they clear that. The first time they got a cold, when they got re-exposed to their family, we just said, well, if their immune system's not going to work, this is going to be the absolute disaster. They're not going to be able to handle that virus or that bacterial infection. And yet, if anything, the patients did better in those next 100 days. And there's lots of reasons we know now why they did better, because they healed more effectively with this immunosuppression. But they didn't have any problem with their first viruses. So there was something that we didn't understand about people. It might not be true of mice, but there was something we didn't understand about people. And so when Carl and I went to the laboratory, we took on as a side project trying to figure out what was going on there. And the basis of 3 years of playing around and combining every combination of ways in which people had said you could induce proliferation in lymphocytes, we found one. Carl really found the combination that worked, that actually allowed human lymphocytes to proliferate perfectly fine with industrial doses of cyclosporin on board. And that pathway is what we now know as the co-stimulatory or CD28 pathway.
During this pathway, or during this time, you were also looking at B cells and T cells and lymphocyte survival.
Right.
which then helped you to transition into the world of apoptosis and discovering anti-apoptotic proteins. I know this is skipping ahead a little bit, but how did you follow this trajectory?
When I first got interested in immunology, and I had my first experience, first in Bethesda, and then second one, I went back and started to work on lymphocytes in my research years at the Hutchinson Center. I really was interested in the problem of proliferation. It fits oncology, cells are proliferating abnormally. Immune cells are the only cells we knew how to put into test tubes and get them to proliferate. The standard assay was that you added a mitogen or your favorite antibody that would stimulate lymphocytes, and then after 3 days you would add tritiated thymidine, and you would wait a day, and then you'd harvest the cells, and you'd count the thymidine that was incorporated in the cells, and that would be an index of proliferation. I got in a lot of trouble right from my very first assay because I had just come from the Naval Blood Research Laboratory where I knew how to count cells better than anybody on earth because that's what we did all day long. And I grew up in a family where my mother was morbidly fearful of radiation. So when we got, first of all, we were never allowed to have a color TV because it gave off too much radiation, but we had a black and white TV, and my mother, every time we moved, put a line 6 feet away, and we weren't allowed to get any closer to that because she was worried that we, my sister and I, as kids, would get too much radiation. So I always thought you should limit the radiation, and so I said, well, I know how to count cells, so instead of adding the tritiated thymidine, which is radiation, I'll just count the cells and how many there are between day 3 and day 4. And many immunologists have actually never to this day done that experiment. And what's interesting is there are less than 10% of the cells that were present at day 1, present at day 4, in any assay that people were doing at that time. So I went to the professor in the laboratory and I said, you know, this is not proliferation we're studying. And he said, don't worry about it. I said, but most of the cells are dead. And he said, well, don't worry about that. That's the trauma of taking them out. And I said, but I take cells out of the blood, which has got to be a pretty traumatic phase. We don't do anything particularly caustic, and we put them into culture and they die. And he said, well, that's just because tissue culture is imperfect. And we kept bumping into that as a problem as we studied the immune system. We couldn't study the phenomena we wanted because the cells would die in culture. And around that time, Stan Korsmeyer, who has been honored many ways here and tragically died young, had just been, along with Carlo Croce, characterizing a gene called BCL2. And the model system that we were working in, to make a long story short, BCL2 couldn't be the example because it wasn't expressed. And so we decided we would go and look and see whether this is a generalized phenomenon and there are other genes. And I actually had my first own technician, and so I set her to cloning homologs of BCL2, and we cloned a gene that now is known as BCLXL, and the rest is history. We had a way to reverse that cell death response. We started with long collaboration with Stan Korsmeyer's laboratory to clone a wide variety of genes in the BCL2 family that regulate this problem of apoptosis. And we ended up getting sucked into the use of that to explain why it is that cells die in culture, understanding what pathways, again, CD28 came back into the picture because it's a major turn on of BCLX, the gene we had cloned, and we probably never looked back from that. We started to begin to become interested in why cells die and how we could manipulate the apoptotic decision to learn more things about cell death and not as much about cell proliferation over the last 20 years.
So during this time, you were at the University of Michigan as an HHMI investigator, and you decided to take on a big leadership position, your first big leadership position at the University of Chicago, heading the Knapp Center for Lupus and Immunology Research. So why start to take on an administrative role when your research is getting so exciting?
I left the Navy and been very fortunate to be picked to be one of the first Hughes investigators at Michigan. But it was a time when science was changing. All the people that trained me in science were actually MDs by training, because the best scientists in biomedicine in the generation before me were all physicians. The microbiology explosion had happened in the period of time that we've talked about, and now the best scientists on earth were PhDs and people that had been rigorously trained the way a PhD was. And I didn't have a PhD. So when I went to Michigan, I was a physician scientist. I had an appointment in the Department of Internal Medicine, and because the Hughes also requested basic science employees for their appointments for them people, I got a secondary appointment in microbiology and immunology. But it was under the condition I would never be allowed to take a graduate student because I didn't know what it would take to be a real scientist, and therefore the department gave me the privilege of being part of that department in exchange for teaching second-year microbiology to the medical students. And I was never completely happy with that situation, and as you said, our science was taking off, and I wanted an opportunity where the science would be the more important part of my position rather than secondary to my taking care of patients with leukemia in the hospital and teaching microbiology to medical students. And taking a leadership position in an institute of medicine where the understanding of medicine would be taken back into the science curriculum gave me the opportunity to concentrate on my science, and it was a good trade-off in many levels.
So, again, skipping ahead perhaps a little quickly, you then took on an even bigger role in terms of administration by, after a time, moving to Penn to help start their Abramson Cancer Institute.
Right.
At this point, your science had also transitioned a little bit more towards metabolism within the cells and more towards cancer cells.
So the offer to go to the University of Pennsylvania and help start a new research institute came at an absolutely perfect time. We'd really completed what I thought I went to Chicago to try and do, to start the Institute of Medicine and the Knapp Center of Immunology Research. We had filled the place up. It was at a self-sustaining level. And I was no longer primarily interested in apoptosis because we had had the fortune to have eliminated apoptosis from mice and found that there was still another mechanism of control of cell survival, and that was through the regulation of nutrient uptake and cellular metabolism. And so I knew I wanted to change the lab to that, and there was no way I could do it because everybody counted on me to be the resource for apoptosis. And they offered an opportunity which everybody sort of talks about. If you could do it over again, couldn't you do it better? So I told the lab 3 months after we moved there that we were going to abandon everything else we did and we were going to focus purely on the regulation of cellular metabolism as it informed cell proliferation and cell survival. Did not lead with a great deal of excitement in the laboratory, and in fact, Jeff Rathmill, whose wife is here at this meeting and was the senior postdoc in the lab, was elected by all of them to come and tell me that they were going to rebel because, in fact, I was destroying all their careers. As you may well know, Jeff's a full professor at Duke University right now who carries on some of the most important work in understanding glucose regulation in non-metabolic tissues and leads the study section for that and everything else. It took some convincing to keep them going, but in fact, all of them now, to this day, work in cellular metabolism.
Okay, so we've talked a little bit about you taking on bigger and bigger administrative and leadership roles. Mm-hmm. And then in 2010, you took on the biggest one so far, and that is as president and CEO of Memorial Sloan Kettering Cancer Center. I know this job well, right? You're leading over 14,000 people now. So how do you manage to yet still churn out such excellent research while you're managing the leadership, the financial, administrative, HR, all of the different puzzles that come along with a job as enormous as that?
Well, I mean, the real secret is to work with really smart and dedicated people who want to actually take responsibility for what they do. And so that's what makes it easy to do at Memorial Sloan Kettering. We all know why we're coming to work. Everybody's very proud and very responsible in their job. So it's more about delegation and making sure we're aligned in the correct way. So I've been very fortunate to work for now over 25 years with my wife, who is better trained than I am. She is an MD PhD from the Karolinska Institute, trained in the Institute of Tumor Biology in immunology, so she has the same interests as I do in oncology and as in immunology.
Now, from your perch at Memorial Sloan Kettering, and the way that you're influencing how that particular institution is looking at cancer. What has changed now over the course of your views of medical oncology as a cancer doctor about the way that we're treating cancer? And what do you see on the horizon?
So I think that we used to think, I have a slide that I use now in a lot of the 1-hour lectures I give, which is the same slide I used back when I first taught students cancer biology 25 years ago, which is we thought that cancer really just arose when cells got stuck in the process of cell division. That was a hopelessly naive view relative to what we know now. And it was informed by one of the really important advances that have happened with molecular biology, and that was the discovery of retroviral oncogenes. And that was the first molecular insight into cancer, and then subsequently the transformation assay that Bob Weinberg worked out where RAS was rediscovered that had already been found in retroviruses. And all those assays, viruses want to steal genes because they advance the viral mission, and the viral mission is to replicate, so anything that gets a cell into replication gives it much of the machinery from the cell to be able to capture. And RAS is uniquely suited as an oncogene to allow cells to live in what were called colony assays. And we assumed that then reinforced our notion that this was the disease of cell proliferation, which the pathologists could see under a microscope. I think the sequencing of the human genome, the ability to fully characterize the wide variety of genes that are mutated in cancer has told us that that's probably a wrong idea about what cancer is. So I think we know two things. One is interesting, and that is that unlike when I entered oncology, we now know that cancer is not a disease that's unnecessary consequences of being multicellular. When I started in oncology, we thought plants got as many tumors as animals. It's not true. There's never been a plant that has died of a cancer. There are many theories about why that doesn't happen. The one I tend to favor is that plants never try and regenerate their tissue. They never try, if you break off a limb, they don't try and regrow it right out of that stalk. They die back, they abandon that. They don't enjoy what we do, regenerative inflammatory repair. In that environment is a very mutagenic environment, and it requires cells to switch from defense back to augmenting repair. It's a very complex problem. And cancer always, we now know from various epidemiologic studies in mammals, arises in that unnatural environment of inflammation. That leads us to very many of the exciting new insights about how the immune system can be harnessed. The ideas of checkpoint blockade, because it's eliminating that natural switch that takes the immune system from defense to offense and repair. And we now know that immune system has been harnessed by most tumors to augment their ability to be successful proliferators. And we never thought about that before. When we think about it from a regeneration point of view, that means that we need in all of our tissues progenitor cells that could repopulate that. And it's led us in the latest issues of sequencing cancer genomes to understand one fundamental problem that had been underestimated in its importance. Those progenitor cells actually have to differentiate into terminal tissue. And that mutations that block its ability to terminally differentiate in an inflammatory lesion will stimulate the cell to keep trying to reach terminal differentiation. And a consequence of that, it will continue to proliferate. There were theories 40 years ago that to be able to differentiate, cells had to replicate their DNA to gain accessibility. That idea is back, but now we have the molecular tools to start to think about it. And if you become completely inflammatory relative to how the world thinks about cancer, there is no cell in our body that ever proliferates. All normal cells in our body are attempting to produce progeny that will repair terminally differentiated cells that have been lost. Whether they're lining our intestine, lining our lung because they've been damaged by smoke, lining our skin because we just got a sunburn. The proliferation of progenitor cells is the attempt to put out progeny that are differentiated. And if you block the ability to differentiate, a normal cell will continue to proliferate. And those blockades are something we have just come to grips with. So we have exciting stuff around how cellular differentiation is really affected in adult organisms. I think that'll be a third of what happens in cancer as we go forward. I think we're going to see exciting new, we're just beginning to touch new immunologic strategies to harness our immune system and our repair systems. So the immune system will be both the innate systems as well as the professional B and T cells, which have created the excitement right now. And then we will see continued success on the targeted therapeutics where we understand the driver oncogenes that do give cells a proliferative advantage and how we can harness them by turning them off to advance cancer treatments. So we're at best at 50% of how we optimally understand cancer and then how we exploit that information in helping patients and their families with treatments. So it's an exciting time right now.
Yeah. Do you remember the first experiment you ever did?
I was given this paper that had just been published in Science by my predecessor in the lab, in which he had worked with a physicist at MIT, and they had discovered that when granulocytes were isolated from blood, so white blood cells are isolated, and you added to the liquid that you suspended them in a LPS, which is a common innate antigen that we now all think about as part of toll receptors, but in fact there are more ancient receptors for them that are part of the innate immune system. Whenever they added the LPS to the culture, the cells sedimented faster in the media, and this was considered the first wedding of physics and biology. We're going to learn this fundamental new principle about how white blood cells could, even in liquid, get more rapidly to where they were needed. And I was told to figure out and design experiments to figure out how this was mediated. And after a year's worth of work, I'd learned enough of the physics and I'd done enough observation of the system. All we were learning about was the difference in the way large particles and small particles sediment to gravity because the white blood cells had aggregated together and there was nothing really interesting that was unknown. So it was a year of, many people would say, frustration and failure, but it was great in teaching you how to convince people who wanted this to be a science paper and more, that it was ultimately a science paper, but the answer to it wasn't that interesting. And so sadly, my very first publication that I was the first author on is published in a journal that no longer exists.
Do you have a favorite book?
And it makes no sense. But the only book that I'm always informed by when I have to think about how to run organizations, which as you pointed out I do occasionally, is Watership Down. It's a children's book written by a veterinarian, but it has every important truth about how groups work together.
When you were a kid, what did you think you were going to grow up to be?
I was pretty sure I was going to be a lawyer. I have no idea why, other than there was nobody in the family that did that, so it seemed like an open field to do it. It seemed like lawyers had interesting jobs. I took one pre-law course in college and realized that was the stupidest idea I'd ever had.
So then a related question, if you couldn't be a physician or a scientist, what other career path do you think you could have excelled at?
So the only other career that I explored, and is actually what I thought I would do until I got into the one medical school I applied to, is I was getting my teaching certificate at Dartmouth. I would have been a high school science teacher and been a sports coach.
Excellent. Well, I think I speak for everyone in being very glad that you chose the path that you did. So thank you very much for taking the time to chat with me today.
I've enjoyed it a lot, Ushma. Thanks a lot.
All right.